So very nice workshop from Dr. Fiona and Dr. Barbara. It was really nice because I am a total user of Campbell and we need your data. We need data from the pharma, and yes, we need negative data. And I'm going to talk about a little bit of our applications and usage of this data for drug discovery or heat discovery for neglected and emerging disease. So first of all, who and where are we? So we are in Federal University of Goiás. My laboratory is called LabMol, Laboratory for Molecular Modeling and Drug Design. And we are located in the middle part of Brazil, in the state of Goiás, very close to Brasilia, to the capital. We are a young university, but despite of that, we have a great number of students and professors. Our building of Faculty of Pharmacy is a brand new building. We renovated it in 4 years ago. We moved to this new building, and my laboratory is located here in the fourth floor. So I invite all of you, when possible, please come and visit us here in Goiás. And I also like to share that LabMol is nowadays part of this center of excellence in artificial intelligence. So it's called SEIA. SEIA is a part of the Embrapa, or referred as Embrapa unit in Brazil. Embrapa is a funding agency in Brazil, which is fostering innovation projects with Brazilian industry companies, Brazilian companies. So it's really nice to be nowadays part of this artificial intelligence center of excellence. So we have many partners and companies that we are developing innovation projects together. So this is really brand new stuff for LabMol, and I'd like to share with you. And our research interests relies on computer-assisted drug design and artificial intelligence guided or oriented design for neglected and emerging disease. We also apply artificial intelligence and computational tools for the development of platforms, in silico tools or platforms for predicting toxicity of chemicals. And this I will leave to Professor Alex Tropsha, who I think will talk about more of some of works that we have been developing together. And today I'm going to talk about more our applications of artificial intelligence and computer-assisted drug design for neglected and emerging disease. So when I started my laboratory, my group here in Federal University of Goiás, back in 2010, we started working with malaria and tuberculosis, as I worked with TB in my PhD thesis at the University of São Paulo. And then we expanded the portfolio for other more neglected diseases, such as schistosomiasis, like Dr. Fiona said, leishmaniasis, Chagas disease, and some viral emerging diseases, such as dengue, Zika, and also nowadays COVID-19. So the drug discovery pipeline, as you must know, is a very lengthy, costly, and uncertain process, in which from the 10,000 compounds that are discovered in the first years or first stage of this process, only less than 20% of those compounds go to the translational and stage and go to the human clinical trial. So it is a high risk process, and we need innovative approach that could bring new drugs faster and a lower cost to the market. Mainly also thinking on those neglected diseases, the neglected populations. Such diseases affect the most poor populations in the world, developing countries and underdeveloped countries. And as we were talking about data, we are living in the era of big data. So there are estimated 10 to 60 chemical compounds that have been already discovered and cataloged. And from them, 10 to 33 compounds which have drug-like properties. And also we have millions of compounds with at least one known bioactivity, 2.3 million compounds in Campbell, 112 million compounds in PubChem. So we are drawing data, but we don't have the knowledge. So we can use this data, the knowledge that have been produced already on the literature, and we can model these, we can develop models to anticipate and to predict the activity or biological properties of new compounds, of better compounds, optimized compounds. And is that we are going to try to do with the QSAR models. So quantitative structure activity relationship is a methodology that is going to relate chemical structures with biological properties. So we are going to build an equation, a model, to relate one or the properties of the chemical structures encoded in the descriptors. So we need a number that can be a chemical property of this structure, and it will be related to the biological property. So we can develop this model using statistical or classical statistical regression or machine learning and deep learning algorithms. And also we are going to validate the models according to numerous statistical processes and domains of application. We can also have classification models or regression models. So in the classification models, we'll have the binary models that we can classify compounds as active and inactive. And in the regression models, we'll have the prediction of the biological property in the end. And what we can do with these predictive developed model, QSAR models. So we can vector screen a chemical database that has never been tested for that endpoint or that disease, that protein that you are looking for new hits. So we can use these QSAR models that were built with data already experimentally produced, so chemical compounds with biological property. We will build those models, and then we will virtual screen a chemical database that has never been. Are you listening to me? Because I just okay.
Yeah, we're listening.
Okay. Thank you. So we can use those models, to prioritize compounds, virtual hits to predict it to be active. And then we are going to experimentally validate those hits. And we can also deprioritize or remove inactive compounds. And we can also experimentally validate the models, testing also some inactive compounds, okay? So this is the main idea. And now I'm going to share some cases that we have here in my laboratory. So for most of those diseases that I presented in the beginning of the presentation, we have models and applications, but today I chose 2 applications for discussing with you, 2 new, very new applications. One of them is regarding Zika virus. So Zika virus is a neglected virus and emerging virus as well. It has been reported for the first time in 1947, and it remained neglected for more than 50 years. And in May 15, it started an outbreak here in Brazil that quickly spread to the Americas, and it was confirmed in more than 50 countries. So here in this map, we can see a roadmap with areas with current or previous Zika virus transmission in 2019. The main Zika virus caused a very mild infection, limited infection, but it has been proven to cause some neurological issues to infants and also to adults. So this is very severe form of Zika virus infection. And also the transmission of this virus is primarily from the mosquito, the Aedes aegypti mosquito, but also it has been found in biological fluids, in blood transfusion, and also mother to fetus transmission. So for those points, the World Health Organization pulled a blueprint list of priority for drug and discovery and Zika viruses in this list. So we had this outbreak in Brazil in 2015, and until today, 2022, we don't have a vaccine for Zika virus, and we neither have a drug to treat the patients infected with this virus. So when we started this project, so I started a collaboration project with Dr. Sean Atkins from Collaboration Pharmaceuticals in the USA back in 2014 for dengue virus. And then when the Zika virus outbreak started in Brazil, as dengue and Zika are from the same family as flavivirus, we just switched a little bit our research and started thinking on how could we start a drug discovery project without data. That time there was no ligand data, there was no protein data from Zika virus. So how could we start a drug discovery workflow for a disease, a new disease, an emerging disease without data? So we pulled together a list of researchers, and we published this white paper, very nice one, with this proposed workflow for quick drug discovery against Zika virus. How could we start with SBDD or LBDD? So we had this idea, and then we started pulling together another team of collaborators, and Dr. Sean Atkins introduced me to Dr. Alexander Perman, and Dr. Alex Perman had a project at the time, a grant in the IBM World Community Grid, which is a philanthropic initiative from IBM, launched in 2004, that provides massive supercomputing power for scientists without any cost by using the idle processing power of computer and Android device. So Alex had this project for malaria drug discovery using the supercomputing power at the IBM. And then we pulled together a project for Zika drug discovery. And this was launched in May 2016, and it's called Open Zika Project. So the Open Zika Project is a global collaboration project, and our main goal was to accelerate the drug discovery of an effective treatment for the Zika virus. And the idea was to use open data, so it was an open drug discovery project. All the data that we produced, it was made open, okay? And we screened all available compounds, purchasable compounds on zinc databases with all Zika virus proteins, which are 10 proteins. And we used this massive collaboration network for computing, for accelerating our computational resources. We have done the massive virtual screening with molecular docking, QSAR models, and molecular dynamic simulations. And at the end, we prioritized some hits, some virtual hits compounds, and then we bought those compounds and performed the experimental validation of those compounds. So I'm going to share one of the examples of application of this Open Zika project, which was recently accepted on Journal of Chemical Information Modeling, and it's just accepted paper. And it comprised the discovery of some experimental hits for the Zika virus proteins, the protease and polymerase. And this work is the result of a 4 or 5 years of research of a huge team of collaborators, starting with my former PhD students and master students. So Melina Motin was a postdoc in my laboratory, and Bruna did her master dissertation in this project. And it's really nice that we can now share these results. So we had provided this massive collaboration network using the World Community Grid network. And then we performed the virtual screening with the whole zinc database to select some compounds for the experimental validation. As our main general workflow, we pulled together the structures of the proteins on PDB, or we generated the 3D homology models for the Zika virus polymerase, the protease, and also the helicase. We also developed in parallel, we developed machine learning models for predicting the phenotypic activity against the Zika virus, the antiviral activity. And then we performed the parallel also virtual screening workflows for each of those proteins using the World Community Grid network. So we started with the molecular docking, then we used the machine learning models for the phenotypic assays. Then we also filtered for log-P, PAINs, and aggregators. And at the end, with a final visual inspection, medicinal chemistry inspection, we selected some of those hits, performed the enzymatic assays, also the cell-based assays, and also performed some molecular dynamic simulations to shed some light into the mechanism of inhibition of those compounds. Regarding the data for developing the QSAR models for Zika virus, at the time we started this project back in 2016 and 17, there was almost no data for Zika virus, the phenotypic data for Zika virus. Then we had this PubChem bioassay for Zika virus, which measures the bioactive of caspase-3 induced by Zika virus infection. So this is an activity assay that measures the cytoprotective effect. It's not a direct antiviral assay, but it measures the protection of the cell to death, okay? So we used this data that was the only data available at that time to start a QSAR model for Zika virus. And then we performed the data curation, integration, and modeling in our developed workflows in mind. And then we performed the generation, we generated and validated the machine learning models using different algorithms, machine learning algorithms, and different descriptors as well in Python. And at the end, we were able to develop models that are very good in distinguish active and inactive compounds for the cytoprotective effect against Zika virus infection. As we can see here, all models, developed models, were had the major statistical result higher than 0.7. So those models were used to separate or to filter compounds should be experimentally validated and to filter out compounds that were predicted as inactive. So at the end, we performed the independent virtual screening workflows for each of the targets. And each of them, we selected some compounds for the experimental validations. And these were performed by the group of Professor Glaucius Oliva at the University of São Paulo in Brazil. They performed the enzymatic and biophysical assays with the proteins, the Zika virus proteins. And the group of Professor Jair Siqueira Neto at the University of California in San Diego, who performed the phenotypic assays, Zika virus cell-based assays, and cytotoxicity assays. I'm going to show you some details of each of these virtual screens, not all of them, because the paper is too extensive, but for instance, for the protease, we performed different virtual screenings regarding the two different binding sites. One was using the PDB with the closed conformation of the protease for using the catalytic site. And the other one with the open conformation with this PDB ID for screening in the allosteric site, for looking for allosteric inhibitors. Then we performed the cascade of the virtual screening and selected compounds for testing for both sites of the protease. When we performed the experimental validations for the protease, we were able to find this experimental hit with IC50 of 7.4 micromolar, and the behavior is like the mechanism of inhibition is of an allosteric inhibitor. We also performed some molecular dynamic simulations to understand the binding mode, to shed some light more on the binding mode of this compound with the allosteric site of the protease. For the polymerase, we used 3 different binding sites for docking. So we used the active site, which is the aspartate residues binding site. We also performed the virtual screening, the dockings on the RNA site, and also the allosteric or any pocket site. Sorry. So we performed the independent virtual screening regarding each of these sites for the polymerase, and we ended up testing the 15 virtual hits that were selected. And in this case, we had more success. We had 5 experimental validated hits for the polymerase, and some of them are in the low micromolar range of inhibition with 0.6 micromolar, 0.8 micromolar. Also, what is very interesting is that some of those compounds were also able to protect the cells from death infected from Zika virus in the phenotypic assays performed using glioblastoma cells. So the first compound, LabMol 301, it was able to inhibit both proteins, the polymerase and the protease, and also to protect the cell from the death induced from Zika virus. We had also other compounds very potent for the polymerase, but they were not able to protect the cell from the death. On the other hand, we had some compounds that were not able to inhibit the proteins or inhibit just with high concentration for the helicase protein of Zika virus, and they could protect the cell from death in the phenotypic assay. So this is very common when we deal with antiviral compounds. Sometimes they can protect the cell from death, but we don't know the mechanism of action, right? And in the end, for this project, we had some experimental hits that were discovered, and this one was the best one that we selected, LabMol 301, that had IC50 on the polymerase of 0.8, IC50 on the protease 7.4, and IC50 in the phenotypic assay for the cytoprotective activity on Zika virus induced cells, infected cells of 6.68. Also, it was really interesting to check the similarity of this compound with other inhibitors of the polymerase of Zika virus. There are just a few compounds described on the literature as inhibitors of the Zika virus polymerase, and they are very dissimilar from our compound. So here we calculated using max descriptors and the Tanimoto coefficients, as we can see. From this one is around 0.7, and all the other ones are very dissimilar compounds. So as a summary of this Open Zika project, so through the World Community Grid Computational Network, it enabled us to perform massive docking-based virtual screening campaigns for all Zika viral proteins. We developed machine learning models for predicting the cytoprotective effect of compounds over Zika virus infection. And we were able to find some compounds that are inhibitors inhibitors of NS5 polymerase, non-nucleoside compounds, inhibitors of the polymerase of Zika virus, one compound able to inhibit the protease and also the polymerase, and 8 compounds that were able to protect the glioblastoma cells from Zika virus infections. So this was a huge, massive collaboration work developed mainly by this team, who I'd like to thank a lot from my team, from LabMol here at UFG. Rodolfo Braga, who is a former lab member, and nowadays he has his own company, a startup called Insilical. Of course, I'd like to thank Sean Atkins and Alex Perman to lead this great project with me, and our collaborators who performed the experimental validation of this work. Then we moved, when we were almost finishing our Open Zika project in the beginning of 2020, then it started the pandemic of COVID-19. And as it was also an emerging virus, Sean Atkins invited me, he mailed me and told me, Carol, we need to collaborate and we need to do something. We need to write something regarding this deja vu that we are living again, a new virus is starting on China. At this time, it was really on the, I think, January in 2020, so it was not declared yet a pandemic. And then we started the team of the Open Zika team. We started pulling together this perspective paper. How could we stimulate the open drug discovery for a new emerging virus? So again, we started with some homology modeling of the protein that, and then we performed a virtual screening of commercial and approved FDA-approved drugs. And in this case, that was very quick publication. We just published a perspective without experimental validation. And then we started pulling together a team of researchers here in Brazil, submitting some grants. And then we approved some local grants with Goiás State grant here in my state and also in the state of São Paulo, PAPES grant with Professor Fábio Costa and Professor Maria Cristina Nonato. And then we started thinking bigger, and we also approved this multilateral project involving the BRICS countries called COVID AI. So this project, our main goal is to pull together a huge, the whole drug discovery cascade for developing new drugs for treating the SARS-CoV-2 infection. So in this case, there is 6 Brazilian different groups involved. There is one very experienced group in South Africa, in University of Cape Town, the H3D Center, Drug Discovery Center, and also the Russian Academy of Science involved in this project as well. So in this COVID AI project, and also we have the collaboration of the company of Sean Atkins in the United States. The collaboration pharmaceuticals is also collaboration in this project. So we started developing the models, the computational models, and in this case we had tons and tons of data for the proteins, for the antiviral activity of. So we had structure-based models, we had ligand-based models. So we developed all those models, shape-based models, docking models, machine learning models for SARS-CoV-2 phenotypic data, and also for the selected proteins that we selected in this project. So this project comprised 4 different proteins: the spike protein, the 2 protease proteins from the virus, and also this human protein, dehydrorotate dehydrogenase, DHODH, which is a protein, human protein involved in the pyrimidine biosynthesis. So it's very important for the viral replication. So we developed all the computational work together with Dr. Sean Atkins in this Open Zika moving for the COVID AI project. We performed many, and we are still performing virtual screening workflows with different models, with different database libraries of synthetic compounds from the other groups, and also from natural products, from Brazilian natural products groups. With the selected hits, we are performing the target-based assays with the groups involved in this project. And then we are also performing the cell-based assays with the virus in Fiocruz here in Brazil, and with the human cell line for cytotoxicity assays. And we are performing the hit-to-lead optimization with 3 synthetic groups, one of the in Brazil from Professor Flavio Emery, and one in South Africa, and other in Russian Academy of Science. Also, we are performing the development of lung targeted nanoparticles for the most promising compounds, and also for some repurposing compounds that we found on those studies. And also we are performing the in vitro and in vivo pharmacokinetic assays for moving to the in vivo models, for the preclinical models on the infected animals. So regarding the machine learning model for the SARS-CoV-2 phenotypic assays, when we started, this is not so new, so we started these in 2020. So we have new models now, but I cannot share yet because they are not published. So this is the first model that we developed back in 2020. We used this data again from PubChem. So we had these compounds that were tested in SARS-CoV-2 for cytopathic effects, and we developed classification models using the best models was using MAC descriptors and random forest machine learning algorithm. So it's not that good model, but was the model that we could build at that time. Again, nowadays we have a better model than this one, but we used this model to screen some database to select compounds. We also performed many other computational work. In this work performed by my PhD student, Sabrina Mendonça, who is working with the main protease of SARS-CoV-2. So she performed validation for docking, for using in the virtual screening workflow. And look at the difference. For SARS-CoV-2, there are many proteins. For the main proteins of SARS-CoV-2, there are more than 300 PDB structures on the PDB. For Zika, almost no structures, some just few structures. So as we can see with the COVID pandemic, we saw the world working together to gain some expertise and to advance in the discovery of a vaccine and the discovery of a drug. And this, I think, was the main main point that we learned in this pandemic. We need to work together to advance the drug discovery, to advance many important fields in the world. So for the protease, we developed the docking models. We validated models using an ensemble docking with 5 different conformations from PDB. We also performed shape-based models with non-ligands for the main protease. So in this case, we used the OpenAI ROC platform for developing this shape and volume complementary. So we validated these with non-inhibitors and inhibitors and non-inhibitors, active and inactive compounds against the protease. So here are the metrics of this validation of this model. And we also performed developed machine learning models for the protease data using classification models and continuous models for using deep learning algorithms. So in this case, for the protease, the main protease, we performed the virtual screening workflow with different databases. In this case here, I'm showing you the results for the screening of the H3D database, which is the Drug Discovery Center in the South Africa partner of this project, which has around 7,000 compounds. So we performed the cascade virtual screening using the validated model for shape-based, for docking, and for the QSAR binary and the regression model as well. And then we selected the virtual hits and we started testing those compounds. And now we have some preliminary results these compounds were tested on the main protease threat assay by Professor Isabella Bastos' group at University of Brasilia. And as we can see, our compounds were not that good. They are high micromolar inhibitor, mid micromolar inhibitor. So we are going back here. We are revalidating some of these models. We are performing also virtual screenings based only on shape-based models and QSAR models. Then we are performing virtual screening based only on docking-based models, so we can see the difference of heat approval of these different computational models and how we can take some advantage from them. We did the same for the other protease of SARS-CoV-2 for the papain-like protease. We had the ensemble docking. We didn't have the shape-based model here. So in this virtual screening, we based only on the ensemble docking and selected 40 compounds. And in this case, we were more lucky, and we could find experimental heat with IC50 of 15, around 15 micromolar. And this was also performed enzymatic kinetics assay, and it is allosteric inhibitor of the papain-like protease, which is also very good. So now we don't have the final result for the COVID AI project, and hopefully soon I will have some results to share with you, some more exciting results to share with you. So I'd like to thank the COVID AI team here in my group at Federal University of Goiás, and also the whole team of the other. Here are only the PIs of this of the teams of the COVID AI project. And recently we had this finally in-person meeting in South Africa. So from Brazil, me, Sabrina, Christy, and Flavio, and the other guys are from South Africa. It was really nice to finally meet them before the pandemic. And my final take-home message is that we need to collaborate to advance something in the world. So as Charles Darwin said in 1800, in the long history of humankind and animal kind too, those who have learned to collaborate and improvise most effectively have prevailed. And also in this cover of Nature magazine, saying that why scientists must work together to save the world. This was published in 2017, and now we see this very huge importance that we need to work together to save the world. So this is my final message. We cannot only do the modeling, we cannot only do the experimental, we need to work together to try to advance in something, to try to save the world, right? And finally, I'd like to showcase our journal, which is my background here. The Artificial Intelligence in the Life Sciences is a forum for rapid open access publication of AI methods and in the greater life science. And I'd like also to invite you to submit your papers to our themed article collection, such as Women in AI, edited by myself, Dr. Melissa Landon, Dr. Raquel Rodrigues Perez, and Dr. Steve Gardner, and also the other themed article collection called AI by Latin America, which is edited by myself and Jose Medina Franco. So we invited you all to submit your high quality papers to our special issues. And finally, but not least, I'd like to thank again the organizers for this invitation. It was a pleasure to be here today. I need to also say thanks to our funding agencies, our collaborators as well, my family, my two joys, my two small babies, and also I'd like to thank all my family here in LabMol, my other sons and daughters, the LabMol group here. And thank you so much for your attention. I'm quite open for the discussion. Thank you.